on. Our next speaker is Pedro Oliveira. We'll be talking about phage defense systems.
So thank you so much. It's really great to be in front of real people after the COVID pandemic. Thank you so much for the organizing committee for this kind invitation to speak a little bit about our ongoing work on anti-phage defensome of ocean, soil, and human microbiomes. So this is how it started roughly 70 years ago with E. coli, with the finding of the first anti-phage defense systems, RM systems, which typically comprise a restriction endonuclease, which readily digests incoming foreign DNA, in this case from a phage, adds specific recognition target sites, and the cognate methyltransferase, which methylates the host chromosome at the same methylation target sites, thus preventing it from being digested by the cognate endonuclease. But this is actually how it's going right now. So being present with so many phages, so many diversity of phages, bacteria have acquired a complex and intertwined network of anti-phage defense systems. So beyond the innate immune systems of RM, we have Brex, DND, and disarm, which also function through methylation. Some of their mechanisms are still very not well known. We have the very well-known CRISPR-Cas adaptive systems, chemical defense, in which some small molecules will bind the phage to inhibit the phage replication cycle. We have abortive infection systems in which the cell will kill itself or arrest its metabolism to prevent the ongoing phage replication cycle, signaling systems, and a plethora of unknown mechanisms such as Gabesia, Shadu, Zoria, for which we absolutely have no idea how they work. So, prokaryotic defense systems and their diversity has been severely limited because most studies focus on the rare cultivar ones. So here, in this work, we shifted to genome-resolved metagenomic data to query the whole defensome, essentially to answer these three main questions, which I will try to unveil a little bit today. What is the abundance and diversity of anti-phage defense systems in complex microbial communities? Can we build upon anti-phage defense hotspots to uncover novel immune systems? And how does community composition and habitat shape the defensome? So what we did was to build upon one of the most comprehensive HMM databases of all currently known anti-phage defense systems so far. So this involves roughly almost 800 HMM profiles targeting almost 60 families of anti-phage systems. And it also relies on very strict organization rules that will demand the classification of genes as mandatory or accessory, as well as a maximum distance between them. So this is exemplified in this scheme. We have three defense systems that are characterized by their sum of genes, maximum distance between them, and number of mandatory genes. So with this pipeline in hand, what we did was to actually query a large cohort of metagenome assembled genomes from three different biomes: soil, ocean, and human gut, in a total of 8,000 high-quality mags from these three different environments. So what did we get? We got around 40,000 defense systems and almost or more than 500,000 defense genes belonging to 50 anti-phage families. Well, RM systems were found always to be the most abundant, presumably because they are the only ones capable of inducing addiction and leading to post-segregational killing. And despite the diversity in terms of abundance and different families, the top most abundant defense systems across biomes, they are transversal, which suggests or leads to suggest that there are common mechanisms of protection. We found that the distribution of defense systems in human and soil mags was roughly normal, whereas in ocean we see logarithmic-like distribution showing that the majority, or a large proportion of that at least, are actually defenseless. We observed a positive correlation of defense systems with mag size. Typically larger genomes engage in higher genomic flux and horizontal gene transfer. And here on the right, we can see that the frequency of the defensome varies widely among bacterial phyla. This is the example for ocean mags. So in purple, you have the density per kilobase pair of complete defense systems, and in red, you have the density per kilobase pair of defense genes. So while no mag was devoid of defense genes, we have plenty of them that are defenseless and others that do contain complete systems, which are typically associated with clades that can engage in a higher genetic flux. So defense cannot be dissociated from mobility. So it means that defense systems cannot be dissociated from mobile genetic elements. So if we zoom in, again, to our example of the ocean mag data set and to prophages, which are one of the most abundant, and if you remember that there was this logarithmic-like distribution of defense systems in the ocean mags, we would expect the same for the number of prophages. And it's exactly more or less what we see here. This is a log scale. So the majority of the defenseless mags are actually devoid also of prophages. But still, we have a positive correlation between the number of prophages and defense systems, showing that there is still an ongoing bacterial phage arms race in the ocean mags, which is much more stiffer, I would say, for more structured environments such as soil and the human gut. And here we found an average of 1.2 prophages per mag. But we did not only look at prophages, we also look at other classes of mobile genetic elements such as integrons, ices and imes, and plasmids. And we found between 30 to 60% of defense genes belonging to 30 different families that are shuttled by mobile genetic elements. So here on the left, you can see that there are strong associations between the number of defense genes and presence-absence of each of these mobile genetic element families. Some of them are transversal across the 3 different environments, such as those in integrons, and others are more variable, such as those from prophages, isozymes, and plasmids. If we look at the fine-grained structure of the defensome, and this is the observed expected ratios according to the mobile genetic element and also the environment, we see that there are cases in which, well, it's very heterogeneous to start, but there are cases in which we have, such as those in Brax, in which we have underrepresentation for prophages independently of the environment. The same for Zoria, in this case for ices and imes, but then others such as Cas are more random. So this means that this is a heterogeneous distribution that is highly impacted by the interplay between defense system family, mobile genetic family, and the species itself. So one of the hallmarks of defense systems is their colocalization in defense islands, which also harbor other non-defense genes. So we also looked at this. This is an example, again, for the Ocean Mag data set. We found around 400 defense islands that we defined as having more than 4 defense systems with a medium DI size of 15 genes. We have here the representation of them, RM, Brax, Cas, Seabass as the more abundance. What was interesting to note was that not all of the most abundant defense systems in ocean mags are actually in defense islands. Saying this the other way around, it's the same to say that some defense system families may favor defense islands to actually leverage inter-DS functional cooperation and generation of novel functions. And here on the right, you can see 3 examples of defense islands found in 3 ocean mags, in which you can see that they have a profuse concentration of different families of defense systems, and they are scattered with around 5–6% of other genes that were classified as non-defensive. So what are these genes? Well, we did some gene ontology analysis, And we found a large variety of non-defense functions, particularly mobility. It makes sense. Many of these genes arrive by mobile genetic elements, but also motility, cilium, flagellum, nitrogen fixation, tRNA methylation. I would say that the $1 million question here is, which one of these non-defense genes functionally interact with known defense systems? And which ones belong to new, previously unidentified DS systems? So in the preprint that will follow up, we'll see that indeed, I can tell you that there are functional interactions between them and that there are new, previously unidentified defense systems. So regarding gene phase variation, just to finish, we also looked at gene phase variation. We know that simple sequence repeats can phase vary by slippage, by deletion formation, by slippage of the DNA polymerase. If these SSRs are located in the ORF of the gene, then this may generate on-off and a switch and introduction of premature stop codons, and also the production of truncated proteins. But if they are located in the promoter of the gene, they may generate a graded expression and a mix of bacterial populations containing a mixture of variants. So we also look at this. We found multiple families of repeats that are overrepresented in the defensome. They are classified here, and roughly 16% of the defense genes across biomes from 8 defense families actually undergo phase variation. This is not exclusive to RM systems. We know very well that several methyltransferases and specificity units phase vary, but we also found evidences for phase variation in other more unknown systems, such as the Zoria and the ZORB gene and the HAM-B from Hashiman. So some of them we know why this is happening. The others we have no clue, absolutely. So the take-home message here is that methylation systems, it started like this. They are ubiquitous in bacteria. They play key roles in defense, generation, gene flux control, and regulation of the transcriptome. Then we move towards the defensome of bacteria, which is a fast-growing network of intertwined systems of growing complexity and often unknown mode of action. And here we use genome-resolved metagenomics to quantify the defensome in a large cohort of MAGs from distinct environments. They are abundant, highly heterogeneous, mobile, phase variable, and co-evolving in defense islands. And coming soon, strong evidence for functional interaction between novel families of genes with known defense systems at defense islands. So I'd like to finish by acknowledging my students and also several collaborators of this and other projects and multiple sources of funding. Thank you very much.
Thank you for this really interesting talk. We have time for questions. I think there was.
Hello, really good talk. I think you talked about the mobile genetic elements that help to move the defense systems around. Have you seen the role of plasmids or like horizontal gene transfer in this movement of different system around different bacteria?
So the question was if there are plasmids that
transfer the defense islands or different systems.
Oh yes, we have found evidence of this, particularly for ISIS and IMS. Yes, so definitely the answer is yes.
Then first back and then here.
Yeah. Okay, it does work. Hi, I'm Nora from Stanford. Super cool talk. I also had a question on the a couple questions on the mobile genetic elements if we have time. But I was wondering if you knew exactly what type of machinery they were using. Like, do they carry recombinases or they transposable machinery? I was also wondering, because I'm not an environmental microbiologist, so this might be a very naive question, but how frequent are these mobile genetic element transmission events in the ocean environment?
Yeah, good question. So I'll start by the last one. So we found much less frequent of, I didn't show those results will be in the paper, but we monitored, we calculated the gene flux, we quantified the gene flux, and we found that this gene flux was highly heterogeneous across environments. It was mobile genetic element dependent, and for example, in the case of oceans, which was the one that I presented the most, prophages were actually the most abundant, but then conjugation with plasmids, and it was very, very, very limited, at least with the MAGs that we studied. And sorry, the first question, I completely lost, sorry.
To rephrase it, because I think I have the right words now. Do you see any types of proteins that are useful in either integrating into the genome or like facilitating that mobile genetic element transfer? Because I imagine that would be like a really rich reserve of them.
I didn't understand the protein, sorry?
Do you, in looking at the mobile genetic elements, do you see any proteins that are useful in like either integrating those into the genome or otherwise performing that type of genetic transfer?
I didn't look at that. I'm sorry. But we can discuss that. Yeah. Yeah.
So I have two questions. One is, do you see horizontal transfer between different classes or, genus, family, or classes of bacteria?
Yes.
Like in human gut. And then do you see enrichment of defensomes, like association with integrons in, say, Enterobacter enterobacterialis, like, multi-resistance in human infections, like especially in, hospitalized patient on prolonged antibiotic treatment. Do you see enrichment of certain of the defensomes and integrons associated with different, like say, families of bacteria in human gut?
Yeah, I understand your question. That was a point that we did not study. So to look at, so the MAGs that we used for the human gut were healthy individuals. So we did not relate that with any type of pathology or clinical setting. I don't know if that was the question. Yes.
Yeah,
yes.
Hospital-acquired infections.
Yeah.
For long-term.
Yeah, that would be interesting.
Yeah,
but
so do you see an enrichment?
I don't know. We didn't look at that. Yeah.
Thank you.
Thank you.
Okay, I think we can take one last question.
Hi there.
I'm Ben. I'm from Cornell. I was wondering if you've thought at all about regulation of these islands, and are they regulated as an operon, or are they individually?
Right. Not as an operon. There is not. I mean, what people know is that they cluster together, and then what do you get there? You have RM systems, for example, which work as we all know how they work. And then you have some interplay, for example, with when you have an RM system with a disarm, they may function together, they may co-opt the methyltransferase from one from the other, but functioning as an operon, I would say no. But I would say that there are a lot of, and we show results that go in that direction. There are much more interplay between these defense systems, but not really as an operon. Thank you.
Thank you very much.
Thank you.
Oh no, I think no.